Hello. Thank you for joining today's session. We are going to wait another few minutes in order to be sure that all the attendees who want to join today can be here. So please stay with us and be patient for a few minutes, and then we will start today's session. Okay, I see that we have more attendees joining. We are going to wait another few minutes to have more attendees joining today's session, and then we are going to start.
I hope I'm not unintentionally on this panel here, because it seems like I'm the only male participant here.
You're more than welcome.
I'm happy to drop out and leave it to the women.
You're more than welcome, Peter. Come on. Okay, so thank you. I see that we have more attendees joining, and we can probably start now. So welcome everyone on behalf of the ISB and the organizing committee. I'm very, very excited to welcome you all to the first session of the 2021 virtual conference of the Society, and thank you for joining. So my name is Federica Quaglia, and I'm the lead biocurator of DISPROT and PED, databases of intrinsically disordered proteins, and one of the organizers of this conference. And before I get into the details of today's session, I would really like to express my gratitude to everyone who is volunteering to help organize this conference that this year we are hosting in our lab, at least virtually. So we plan to have some more virtual sessions during the year. In October, as you probably already know, we are going to have a poster session along with the ISB general meeting and a panel on the history and future of the society. So please continue to submit your abstracts during the next month using the conference registration form that is available on the ISB website if you want to be considered for a talk or to present a poster. And in October, we are going to use GatherTown for the conference in order to simulate an in-person poster experience. So please stay tuned for more information. Today's session will include a workshop and invited speaker talks. So in the first hour, we are going to have a workshop on equity, diversity, and inclusion that is going to be chaired by Sushma and Naithani. And in the second hour, we will have 4 speakers who submitted their abstracts, and they are going to talk about their work. The talks will run for about 12 minutes each, and we will have 3 minutes for questions after each talk. So please post your questions using the Q&A box in Zoom. I also suggest you, if you are not already a member of the ISB Slack, to post to join the workspace, and I will post an invite now in the chat so you can see the link and access the workspace of the ISB. So now I will hand it over to Sue for a short demo on GatherTown. So you're going to see what we are going to do in October. So please, Sue, you can.
Thank you very much, Federica.
You're
welcome.
welcome.
I'll share my screen. So we took some time to explore different apps that would let us have more of a poster experience and less of the like 5-minute Zoom poster. And so what we had settled on was this GatherTown, and you will send out links. And when you come, you'll get to something like this, and you can log in and you can, waste time customizing yourself, and then you can join a gathering. This current one is just their standard setup for a conference. And when you come in, you enter a room, and Sandra is very kindly my demo person for another person in the room. And you just use your little arrow keys to walk around, and there's places to meet and talk. But the reason we wanted to try this was, and I think we will redesign this before we do this, is that you can actually, in a poster room, have posters up, and you can get the same sort of experience you would get in a poster session, where you can walk by posters, and I'm having some issues with things uploading, but as you walk and approach a poster, you get a little preview of what the poster is about. And then if you hit the X key, you can actually pull the whole poster up, and you can see the poster and you can read the poster. And then if somebody else is in this same little space with you, you can chat with the poster presenter just like you would at a regular meeting. You can walk by the posters and go, oh, that title looks interesting. No, I don't want to look at that one. Yeah, all the rest of these are just demos. And then if you're looking for another participant, there's a list of participants. You can talk to the person next to you if you're both, or if you're in the same private space. Right now I have my video and mic muted to avoid feedback issues, but you can chat with people, so you can actually talk to the poster presenter. You can, if you look for somebody and you click on them, it will draw a little line so you can go find them on the map. And if I wasn't standing right next to her, the line would be longer. But it gets you closer to an in-person, so you can spend as much or as little time chatting with each poster presenter as you want. Assuming that we go for the full day, if we have enough poster interest to justify the expense, you can actually have the room up all day. And if we have enough people from enough different time zones, we could have rolling poster sessions so that the people in your time zone, you could actually interact with them at a reasonable hour of the day rather than at 2 in the morning if you're in Australia or Asia. So that's our plan. Hopefully that drums up some interest in doing this. One of the nice things I noticed is that while the poster has to be a JPEG or a PNG, it doesn't really mind what size it is. So I have, like, this poster is more of a vertical and this one's more of a horizontal, and that's fine. So you'll have some flexibility in what you want to do. So if anybody has any questions or suggestions for things they might want to suggest we try, if anybody has any experience with this, we'd love to hear. And that's all I really had to say about that. So I will hand it back to you, Federica.
Thank you, Sue. And thank you for the very nice introduction to GatherTown that we are going to use in October. So now we'll hand it over to Sushma, who is chairing the workshop today. So the workshop is about equity, diversity, and inclusion. And Sushma is an associate professor and biocurator at Plant Reactome. So please, Sushma. We cannot hear you.
Hello all, and good morning, good afternoon, good night, wherever you are. Welcome to ISP conference. I'm just going to share my screen for a minute. We are having a workshop on equity, diversity, and inclusion. This workshop is organized by the EDI Committee of International Society for Biocuration. Earlier in the summer, we hosted a movie, Picture a Scientist, and this workshop is a follow-up of that. Although Picture a Scientist mainly talked about the gender and racial bias. We know that there are many different types of bias. If I see the cognitive bias index, there are at least 180 listed there. We are not in a position to address all of them, and we cannot solve the problems related with all of them. However, as also in the movie, we see that there is an implicit bias or unconscious bias, and all of us might have some of these. And one of the goal of this workshop is to be aware of it and talk about these and how people who don't intentionally commit a bias can be aware of some of these things and correct their behavior, attitudes, and practices. So So today we have 3 panelists in this workshop, and I welcome all 3 of them. So we have Vashta Kodia, we have Yasmin Faruq, and we have Laurie Goodman. So welcome to you all. Before I start the workshop, could you please everyone introduce yourself? I introduce myself. So I'm a research associate professor at Oregon State University. I have been working as a biocurator for Grameen database, mostly for the plant Reactome portal, and we curate pathways and genes and also for the various plants. So Jasmine, please introduce yourself.
Hi, everybody. I'm Yasmin Alam-Faruque, and currently I'm a senior biocurator at Healx. And previous to that, I worked as a scientific data specialist at Eagle Genomics. And preceding that, I was a biocurator working in the UniProt, Intact and Goa databases at the EBI. So I've been a biocurator for quite a long time now. My previous experience before that, I was mainly in academia, doing a postdoc in dermatology. Then before that, a PhD in cell and molecular biology. And I also have a master's in immunology, looking at corneal allograft rejection. I also have a postgraduate diploma in instrumentation and analytical science, following a degree in biology and chemistry.
Laurie?
Hi, sorry. I'm Laurie Goodman. I used to be the editor-in-chief of GigaScience journal. We've become a formal press, so I am now the publishing director of GigaScience Press. And we publish at this time 2 journals, GigaScience and Gigabyte. These are both based on large data. And so we have, as part of our staff, biocurators, to help authors organize their data, as we require that data be openly shared for anything that's published in our journal. But for usability, you require that. So I am not a biocurator, but I am tangentially aware of the importance of biocuration. And I run a Women in Science conference every year in mainland China. So this is my general involvement in both curation and more specific in women in science.
Hi, Varsha.
Hi, I'm Vashta Kodia. So I've been a Biocurator for almost 20 years now, starting in the Human Gene Nomenclature Committee back when the chromosomes were being finalized. Then moved into gene ontology, annotation, so did that for quite a number of years. And then I found myself in the world of scientific publishing, which I've been in for the last sort of 8.5, almost 9 years. So started at the journal F1000 Research, where I was data publishing manager, moved to scientific data, where I was the data curation editor, and latterly data curation manager for Springer Nature, which I'm just finishing. So in September, I actually start a new job with Health Data Research UK. So looking at helping researchers manage their data and figure out how to access the sometimes challenging data that we have when we're talking about clinical data sets. But through all of that, I think the kind of understanding of how data is managed and how to curate it to make it understandable has kind of been a thread right the way through. So I'd say I've been basically in curation all of my professional life, and really pleased to be here.
So so the way this workshop is going to be, I'm going to pose a question, and then we are going to have the response one by one from 3 panelists. At the same time, we are also having an anonymous poll. So if you see questions, please answer that. It is anonymous, your response is anonymous, and help us to understand the context and our workplace and the issues related to EDI. So I have a first question. As I said that earlier, we hosted, EDI committee hosted Picture a Scientist movie. And so my first question is that, have you watched this movie, and what is your reflection after watching that movie? So how about So anyone who like to go first?
I can go first.
Yeah, sure.
Yeah, I mean, the message I got from the film was that we should, stand against harassment and discrimination. And however, it's not easy as a woman to be believed when you are harassed. And that highly positioned individuals are favoured more by certain establishments, be it that they bring in money to the institute, than the victims of this kind of abuse, and that there's still an imbalance of women in the scientific faculties, and even more so for women in colour, which is, it's kind of like a given, like, that there aren't that many women of colour in faculties. And there's many issues that, came up within that film that resonated with me, and I'm sure with all of us. So I'd like to, that's my take-home message from that we should fight and do more to resolve the harassment and the unconscious bias issues that come up in society, and especially within the scientific field.
Thank you. Very insightful. Laurie, you would like to say something?
I also saw it, and I asked my sister, who was a lawyer, which is also a predominantly male community, because I was interested in the difference between scientists and the issues that other fields have. First of all, when I saw the movie, I have to admit that I cried in a lot of places. It exactly hit nearly every single thing I experienced when I was at the bench. And one of the most resonating points was that you are not in a position of power. And so whoever is above you, if you are stopped in any way, if you are disliked by them, if you try and get them in trouble because they've been harassing you, you can completely have your career ruined. And because in the sciences, you really have nowhere to go if you can't get through your PhD. You would have to start all over again at somewhere else. So it's more than just fighting and all of that. It's how do we get past the complete loss of progress when we are blocked, and what types of strategies do we have? My sister, who's a lawyer, said that she was shocked at the low number of women still in science, because women have made progress in law. And she said one of the things is you are not blocked early on. There are a lot of places that you can go where you're not starting all over again. But also because clients are now requiring diversity to go to specific law firms. So it's more than just inside the law, it's also out. And that requires, for us, universities and funding agencies to take a stand on this sort of thing. So basically money does what it's coming down to. So very depressing, completely my life, and interesting in the perspective of why things are harder for women in science than in other male dominant careers.
Thank you, Laurie. Very insightful. I agree with you. And I think most of us resonate with that experience as well. Varsha, what is your take?
Yeah, I mean, agree completely with what Yasmin and Laurie have just said. I think my main thing that I took from that film, though, is that it takes really brave individuals to stand up against the status quo at the risk of your own career. And so I think, the fact that, the scientist who was getting harassed at the Antarctica, she didn't feel able to raise a case until 17 years after the fact, once she had her career established. And that's really telling, and it shows her bravery and courage to just keep going in the field that she loved, despite the experience she'd had. And I think what we see is it is survival of the fittest is what we see, and that pressure is not put on men, and it is very much put on women. So what we see at the top are the women who've actually just got the gumption to keep going. They've got something about them that helps them, whether it's that they found some way to have have friendship group or peer group within science that can help them, or they've managed to find a mentor. But for men, this isn't. I think it was really telling. The guy that helped, I can't remember, Adam Lewis, and he said he was there. He was there in Antarctica, and he didn't see what was going on. And I think so. The real take home for me is that it's not just the people who are experiencing the discrimination that need to stand up. What we need is we need allies. We need people who see and notice what's going on to then stand up, because it's a very lonely place to be experiencing discrimination, and to be able to stand up against that, it's very difficult. So I guess take home, find allies, and for those allies to notice what's going on and be vocal.
Thank you very much. I think for the people who are people of color and immigrants, there is another dimension. And the dimension is that your visa and your stay in a host country is tied to your employment. And so not only you are taking a risk on your job, you are also taking a risk on your visa and all kind of the the dominoes effect that comes after that. So it is kind of difficult situation in that sense. So it multiplies, it adds multiple layers. So I think that one of the things we have in sciences is the recommendation of your previous supervisors, and it counts so much. And unless you have the position when you don't depend on the recommendation, then only perhaps you get the courage to stand up. That was another thing, that became very clear by watching that movie. So the thing is that, are you aware of any strategies or any mechanism that can help the women in sciences to solve some of these problems, and what can we do?
I'm happy to talk. I'm very opinionated on I'm opinionated on everything. One of the biggest things that they've found statistically is if young women get a large grant, their career trajectory is very similar to men's. I hate always talking about money, but money is one of the drivers for every single group that has disparity. And I think that one of the things, and it's one of the things I do, is training young women on how to write really good grants can really help. I think this is one of the things that is definitely important, and money unfortunately talks. And if you have money, you also have your own power. So I would definitely encourage both societies and institutions to do specific grant training, sorry guys, for women.
I'd agree with Laurie. I think money does talk, and I think I would go, so absolutely that's helping women to kind of help themselves, but I think also taking away grant funding or taking away positions from people who've been shown to harass or discriminate people in their groups. I mean, I think there's been for a long time there's been this culture of, well, this person brings in so much money, so we'll just ignore all of this other thing. I mean, it's, as it said in the film, it's easier to drive this one woman out of science than to let that man go who was bringing in lots of grants and was obviously very successful. And that culture also needs to change, and that has to come from institutions. It has to come from funding agencies who want to make a concerted effort to change the culture. I think the other thing about science is that you are so, it's unlike any other field where you choose a lab and there's probably one or two people in the entire world that can train you to do the kind of work you're trying to do. And so it really is detrimental to your career if you wish to go into a particular field and you have a blocker like that. There is no other way around it. I think in the private sector, you've got HR, you've got other teams, other groups you can join, other companies you can join. And so it's not quite the same kind of blocker as you get in science. And I agree with Laurie, if you don't sort of get your career up and running early on, it just becomes progressively harder and harder to actually have a career in science.
Thank you, Varsha. Yasmin, do you want to say anything?
No, I agree with both Varsha and Laurie, but I think another thing that is that I think what we have at Healx, we have like a specialist interest group for diversity, equity and inclusion. We have a people team like a wonderful HR and people team who are, very friendly and approachable and have a neutral, receptive view towards all of the employees, so that if someone is being harassed, you can actually feel comfortable going to someone to report that harassment or inequity. And we also have, I think this is like an industrial thing, where we have a regular survey called PCON, which is used by many high-tech financial industries to ensure satisfaction of their workforce. And there are questions built in there about diversity, equity and inclusion. And so if anything does come around harassment related, it's reported anonymously initially, but it's then acted upon quickly by the people team and the line managers. So I think, there are things that people or, different industries can adopt to help with reporting harassment.
So as we said, what can institutes do? So one thing is that training the individuals for getting more funds. And we know that before you finish your PhD or postdoc, most often we don't have that privilege to write independent grants. So training women in grant writing is a great idea, and perhaps it can help them to secure positions. But it's not going to solve the problems of the younger scientists who are in the graduate school or who are the postdocs or funded on the projects and so on. And other reality is not everybody is going to get the grants also. So this is only going to solve part of the problems. What can the institutes do? What are the mechanisms that you are aware of are in your institutes? So for example, in our institute, from past 1 year, we have a more focus on equity, diversity, and inclusion. And we have gone through a couple of workshops where somebody comes to us and somebody who's trained in diversity issues talks about the issues. So education could be, one way to make people aware of it. And socially, I think we owe it to the Me Too movement a lot, that the issue of the women and gender has come into the focus in past years. So, I recognize the value, what is happening in the society will also perhaps influence the sciences as well.
I think one thing, so similar to Yasmin's described, having like HR have a whistleblowing place where you can just go and anonymously lodge a complaint that will then be taken seriously would be a really helpful thing as a first start for any research institution to set up, and to make sure that when new PhD students are onboarded, they know that this is something that exists. There's no point in setting something up and then not letting the people know that it's there. So if you get every time a PhD starts or a new postdoc starts at the institution, to let them know that these avenues are available. And then I think it's also a case of making sure that your tenured professors are getting ongoing training about unconscious biases and being aware of appropriate and inappropriate behaviors towards their students and their postdocs, and generally the people they're responsible for in their labs. So I think that would be my suggestions for what research institutions could do.
Laurie, do you like to add anything? Yasmin, would you like to anything to this question?
I think I just mentioned like some kind of union approach, like Varsha was saying, some kind of HR where people could go to and feel comfortable going to report harassment.
Yeah, thank you. Anybody like to add anything else? Okay, so the movie was mostly on the racial and gender discrimination. We know that conscious bias runs deeper, and some of us and most of us perhaps are not, we don't face the sexual harassment. Actually, I have not faced, I can say that I have not faced the sexual harassment, and but then there are a lot of unconscious bias that we have faced multiple times, and I think it is equally damaging. And then movie also talks about, and as I showed, that it is only the tip, the sexual harassment and racial harassment is only the tip of the iceberg, and there is much more things that are below it, and we have many multiple type of the unconscious biases. Some of we are aware of, some of we are not aware of, some of we only feel when we are subjected to and we are on the receiving ends, other we are very blind. And I think perhaps education could be one way, and talking about these things, addressing those, finding the solutions, believing that people are not biased consciously, and they actually like to improve their behavior if given a chance and given the proper resources. So it's you know, one of the things is that unconscious bias can affect gender, it can affect sexual orientation, your ethnicity, how you speak, your accent, how you look, how you dress, multiple of those things. And so one of the attempts is to do the to bring the awareness and have some educational resources. What roles can societies like International Society of Biocuration can play in such direction within the community? And what is the take-home message we have for the biocurators?
I'd like to add something for the implicit bias. This is something that actually scares me because it's very difficult for people to even realize they're doing it. Just because this is something that a lot of times is embedded from very early on. So you don't even realize you're doing it, much like was mentioned in the film that her colleague didn't realize what was going on. I'm putting a link in for a set of tests. It was also mentioned in the movie of assessing your own implicit bias. And you can take tests for a whole bunch of things. And it was fascinating. I've taken a whole bunch of these tests, and my bias for careers is that I'm moderately biased in terms of men having a career over women. But in sciences, I'm moderately biased in women having a career in science. And I think it may be because I've been really focused on promoting women in science. And a lot of times, if I'm asked to vote for someone to be in a particular area, I will select a woman over a man anytime I see that, as long as I know that they're relatively close. And so I wish that I could take this test farther ago and see how things have changed. But having people take a test like that as part of the discrimination education is, I think, would be something that is essential as part of the discrimination unconscious bias education, because finding that out makes me want to look at how I'm doing things. So I think something like that should be included to address that issue.
It is very interesting, actually. I took also some of these tests, and as a biocurator, we pay so much attention to accuracy and entering the data. It was funny taking those tests. So I think it is geared for people who are not day and night doing, something like a data entry and biocuration and paying attention, synthesizing information. So for me, it was really very funny experience. Because it, but these are a good resources, good tests. I think that, yeah, I agree with you, Laurie, that recognizing even our own unconscious bias is very difficult. So like 2 years back, I took a diversity workshop at OSU, and there were many exercises, how people interpret different behavior. And one of the things is that in Western culture, you have to, when you speak to someone, you have to speak eye to eye. And it is considered as a sign of honesty and confidence. In Eastern culture, in many Eastern cultures, not all, you look away, and it is a sign of respect. So in West, it is sign of dishonesty, and in some other cultures, it is sign of respect. And looking straight is also very offensive in other cultures. So just because we also interpret our body language, how we see things, may influence what we think about other people. So bringing the awareness from multiple dimensions is also very important. The other funny thing is that we were listed whom we think people belong to which groups, and then self-identification versus what other people assign you the identity was something very eye-opening. Some of those things. So this is, I think, a continuous training, and we have to be continuously aware of it. So I thought that training was actually very effective in opening my eyes and people who attended with me. What are the other mechanisms that are effective in addressing harassment, unconscious bias, and so on?
I think again, having a good HR department where the people who have been recruited to be the HR personnel, you have to make sure that they're the right type of people who are, not easily biased towards a perpetrator who may be of a higher status than the victim of the harassment or inequality. And then provide a cultural or environment that is open and friendly and respectful, where people feel valued and where bullying will not be tolerated under any circumstances. So I think, there's, there is an opportunity for these mechanisms to be put into place even in the biocuration field, because it's mainly academia that hosts biocuration, although it is changing now towards more industrial and publishing areas as well. So yeah, that's what I think.
I would go down to we need data. So I think someone asked in the chat about, do we know, for example, in the Biocuration society, what the gender breakdown is, the balances of our membership. But I think it was really telling from the film as well that the data is what was needed in order to change an institution. And so we have to be prepared to collect that data and to be reactive to what it shows us. So if it shows us that women are being paid consistently less than men, we need to then take that on board, take that sigh, that breath, that, oh my gosh, it's really true, and then do something about it. So I think, yeah, for me, it's like we just need to collect data on all of the different protected characteristics. For example, in the UK, we have several different protected characteristics. So these are the kinds of things that you would be relatively easy to collect data on in the UK, for example. And I'm sure the same would be true in other parts of the world. But I just wanted to also just go back to Sushma, what you were saying about culture. And I'm just going to share in the chat. This is a book that I've just been reading, and it's talking about how culture changes across countries. And it's very much looking at it from a business angle, but I found it really instructive. And being a person of South Asian descent, being brought up in the UK, kind of being across two different cultures, to be fair, right the way through my, childhood, schooling and professional life. I found that book very interesting. So Sushma, like you were saying, in some parts of the world, looking someone in the eye is the right thing to do. In other parts of the world, not looking someone in the eye is the right thing to do. And just understanding that difference based on where different people are coming from. And the book is really good because it goes through so many different types of things that you might want to consider depending on who you're speaking to and understanding where they're coming from. And I think that if you can understand where people are coming from, it just inevitably means that you become a more tolerant person and you're more accepting of differences. I think that's the other part of it. Just to be accepting that someone may have a different journey to you, and that what that then means practically, again, going back to the book, women have a headwind that men don't even recognize. And so a woman of color has two types of headwinds that, you might not recognize if you aren't a person of color, for example. So these are the kinds of things to maybe think about, and just if each individual makes themselves more aware of differences, it's going to result in a more tolerant culture. But in terms of changing things on a structural level, which is, I think, what needs to happen, you just need that hard data to show the facts that there is there's a discussion happening here. Here's the data. Now let's do something about it.
Thank you, Varsha. So following your point, I guess that the societies like ISP can perhaps collect the data on gender discrimination, racial discrimination, resource distribution, a number of the women getting and heading labs and getting the grants. And publishing such reports could be one of the, and gathering that data could be society's activity, perhaps. And Laurie, you are the editor of the journal. Would you be willing to publish such reports or encourage a few things like that?
Yeah, we actually do a lot of times publish commentaries and also longer things, reviews relating to a variety of areas that impact the sciences. And one of the things we really focus on is getting diversity within our editorial board, looking at our reviewer profiles to work for diversity, not just men and women, I mean, the balance of men and women, but different countries that are typically underrepresented and all of those types of things. And so publishing something like that would be of interest to us.
Yeah, thank you. Yasmin, you have anything else?
No, I think everything's been covered. I just think, yeah, the ISB is a great resource of data, like Varsha was saying, and it's really important that we do gather all that data and have somewhere to publish it, because that will also, set a, for example, publicizing salary ranges, you'll set a benchmark for other industries to follow. And, you can see if there's a discrepancy between your male colleagues and female colleagues. And, as more industries are realizing the importance of biocuration, we would have an infrastructure to enable, reporting of this inequity as well. And all those, in terms of legalities and policies that will have to be put in place as well. I think it's important that the ISB considers things like that as well. And I think the ISB is big enough to have enough resources to enable this and then publish as well.
I have another question, and my question is that, historically, there have been more women who were biocurators, and because this role also helped them to have the work-life balance. And in it, and not many men actually sought into biocurators' position. So perhaps most of the biocurators, we don't have the problem of the ratio of less women in biocuration. In fact, there are more women in biocuration. And with this particular group, what are the issues that we like to address beyond, the gender bias, or beyond the ratio of male versus female? We have many female supervisors and many senior curators who are the female, and how that mentorship or relationship or having more women as a biocurator has influenced the society.
So in publishing also, there are more women than men in publishing. However, when you start looking at the top of the publishing companies, you see more men. And I don't think that's the case in the ISB, to be fair, but that's the thing to maybe think about, is how people progress up the ranks. I think the other thing that has always struck me about curation is it's kind of a service role. It's a kind of supporting role for research. And it seems to me that those kinds of roles, again, attract women, or end up having more women in those roles than men. It's, like when we think about medicine, surgeons say tend to be male and nurses tend to be female. And I know that's changing, but that's what we see. And I think we see that across all levels of society. So I wonder whether there's something within that where supporting roles and kind of doing, you know, if you think about the hunter-gatherer thing, men tended to be the hunters, women were the gatherers, but it's the gathering that actually enabled the survival of the human species, right? And it's somewhat similar in, I think, in curation. You know, we talk about all this data science, but we can't do anything without good data. And who's cleaning the data? Who's managing this data? Well, it tends to be curators who tend to be women. So I wonder if there's elements of there that is something that the ISB could also investigate and think about in terms of, you know, I've often heard curation is undervalued. Is it undervalued because it's kind of almost like a background thing? Is it undervalued because it tends to be women that do it? I don't know the answers, but I'd be interested to find out.
Jasmine, would you like to add anything on that?
Yeah, I agree with what Varsha said. I think, yeah, it is mainly women, and therefore, you know, it's like, you know, are women paid lower than men as well? And, you know, are we accepting of that? It's just all those things that, you know, come into play, and having a break from science in particular, to have a career break, to have children, impacts your ability to go back into research. And therefore, you know, for me, biocuration was sort of like a backup, because I couldn't get a job back in the lab doing research. So it's not like biocuration is a second-class citizen. It's just that was the only available position that would take me on after having a career break. And, you know, therefore, you know, even though biocuration is kind of like a service, you know, service activity, then, you know, people in service, there are some people in service who get probably paid more than biocurators, you know, underground train drivers, for example. So, you know, it's kind of those kind of comparisons that you have to make. And we are highly educated people, who you know, do the biocuration jobs, because it does, you know, require that attention to detail, and understanding of science and biology and all other aspects of, you know, chemistry, or, you know, whatever else that we're involved in. So it's really, you know, those kind of things that we have to be aware of as well.
And then there is also the job instability. You know, the tenure professors or people who are on the tenure track, or people in the industry, they have relatively more stability, or biocurators have the more stability. What is the journal thing? Because I think biocurators are always paid from the grants, and that are for, you know, 2 to 5 years. So there is a lot of instability as well. So we have a couple of questions in the chat, and I like to take some of those. We have 10 minutes. So let's take 3 questions. And one of the question I have is that from Andrew Sue, is that recognizing that implicit bias is real, I have heard the argument that there is no strong evidence that implicit bias training produces lasting change. So really the training, does it do anything, or institutions just, you know, organize a couple of workshops and get away and tick mark their DEI thing? So what do you see about that? Is it going to have any impact or not?
I'd agree with what Andrew has said, and I think what we have to do is we have to keep our institutions, we have to ask them to remain accountable to us. So again, coming back to data, you know, can we see the data on how, on the, I don't know, the gender of people that applied for a particular job versus the gender of the people that eventually got that job, for example, and got along, you know, ethnicity lines as well, whatever is relevant for that role. And I know it's tricky because sometimes you've got very specific roles, but as a whole, we can say, you know, I don't know, we had 100 job applications over the last year. This was the diversity of the applicants. This is the diversity of the hires, and maybe compare those kinds of data and ask for those kinds of data. I think institutions won't won't share that type of data unless they're really, really pushed. Again, from the film, wasn't there some type of data that the professor had to ask for 3 years to get access to the data on gender salary, right? So you have to be really persistent, and you have to want to make a change in order to make your institution also want to make a change. So I totally agree with Andrew. You see lots of, especially I think since last year, diversity, equality is it just everyone talks about it all the time since George Floyd, right? But what does that actually mean? What's that going to translate to in practical terms? I don't know, and I'm a little cynical of that too, but that doesn't mean we don't try for that ideal scenario that we wish to see.
Laurie, do you have anything?
I just wrote in the text that
yeah, I see that. Yeah.
Yeah, I mean, we talked about how do we protest this, and as she said, we're not going to get that information. But if external organizations were to require institutions, external organizations like funders, charities, et cetera, were to require an audit from institutions to look at level of your job and how much you were paid, then it would become very clear. It's very difficult to do that from the inside. And obviously a lot of institutions would say no, but as more and more institutions say yes, it does put pressure on changing that process. So if funders say, we won't fund people, we won't fund your university unless we have information on this. So, this has to come from outside, and there's no way that it would be able to consistently come from inside.
It is so difficult to fight the status quo. And so there has to be, I think, I see that as a positive sign that at least we are talking about something. Earlier, women were suppressed and other people were suppressed from even talking about the issues. So at least I think I see that as a positive, that we have made a progress, that we are ready to talk about some of these issues. But it will take time, hopefully. We have another question that is going about the titles of biocurators. So the titles like data engineers, biocurators, senior biocurators, lead biocurators, things like that. What does it really mean? And can it help the biocurators community to have some kind of these standardized titles and ranks and some kind of the experience, valued experience associated with each title?
I would say it's imperative that we do have that kind of structure, because going into industry from an academic or service biocuration field and going into a small biotech, even though I had 9 years experience of being a biocurator, I was not allowed to use the word senior biocurator when I joined the company, and I had to change my title to something else, like a specialist data specialist, scientific data specialist or something like that, which didn't tally with me being a biocurator. And only now, working at Healx, have I, got that title of being a senior biocurator. But I think it should be, it's really important to have that structure that has been defined by, societies like the Institution of Biocurators, because then industry will also be respective of that, and they'll understand that there is this, career progression within biocuration as well.
How about the impact on the grant, federal or other granting agencies? Because otherwise they will just think, all biocurators put in a fold and basically give less money, and then PIs who receive that money, they are also kind of pressurized to minimize their salaries so that they can fit into the grant structure and something. So I think it is important also to communicate that different experience brings different value. Biocuration is also layered, and it has hierarchy in terms of the kind of the information different people curate and organize or they develop as a professional. So it is good to have some kind of the structure, professional title structure and salary structure for that.
I think just to follow up on what Yasmin just said, I think actually that would be a really great output from the ISB is just that kind of what does a starting curation post look like? What does it look like to be a bit more experienced? What would you expect from someone who's got 9-10 years of biocuration experience, regardless of title, but just a description of what that progress looks like? And that would really help. That's something that I certainly saw was missing, and I think part of it is because curation is still a really novel kind of career. I don't know, we've all been curators for like at least 20 years or more, but it's still novel in the grand scheme of things. And I think that's part of the issue for curators in general is that recognition, because there's not an understanding of what a curator does, what a curator can do, what that leads to, and that's partly being defined by societies like the Biocuration Society, but putting out kind of like say white papers or something that makes that a bit clearer for everyone who might be interested in hiring a curator would be really useful and helpful.
So we have now 3 minutes left. If something that I have not asked you and they were not the direct questions in the polls either, I'd like to give each of you 1 minute to summarize and thank you all of you and all the participants of this conference.
So thank you, Sushma, and thank you to the panelists for such a great discussion on a relevant topic. I see that we also have some additional questions in the Q&A, but you can also type answers there and keep the discussion going on in the chat. So we will now be moving on to the next part of today's conference. We now have the 4 invited talks from our speakers. Each talk will run for about 12 minutes, and then we will have 3 minutes for questions. So please type the questions in the Q&A box, and we will take care of them. So now I would like to introduce the first speaker of today, who is Nicolas Miliaras, and he's going to talk about identifying new chemical and genetic terms for inclusion in the MeSH vocabulary. So thank you, Nicolas.
Thank you. Just share my screen here. Can you see me? I have my video on, I think.
Yes, we see you.
Okay. All right. Well, good morning, good evening, good afternoon, everybody. I'm Nicolas Miliaras. I'm a technical information specialist at the United States National Library of Medicine. And today I'm going to talk about how we identify new chemical and genetic terms to include in the MeSH vocabulary. So I'd like to start off with the question of what is indexing and what is an index? Trying to advance to the next slide here. Oh, there we go. So an index is, as you may be familiar with, a guide to facilitate the retrieval of content. And you've all seen at the back of textbooks indexes. That's an example of a closed index. And indexing is the process of creating an index. In the case of closed indexes for books, this is extracting key terms to put in the back of the book index. However, for a collection of literature, such as scientific articles published in PubMed, this involves what's known as an open index, which is continuously updated. And open indexes are also known or can be represented by controlled vocabularies. MeSH, or medical subject headings, is the controlled vocabulary used by the National Library of Medicine for indexing literature published in PubMed. And MeSH is a hierarchical vocabulary organized as a tree, which I'm showing you here in the MeSH browser. It has these different categories of the subject headings. And as you can see, it covers everything from anatomy to geographicals, information scientists, science, healthcare, a broad range of categories. What I'm going to focus on here is in the D tree. chemicals and drugs, as this includes most of the new terms that we encounter in index in the literature, such as compounds for drugs, natural compounds, also proteins and microRNAs, gene products. So as you see here, it gets more specific as you move down the tree, different groups of chemicals. And if you look at if you search an article in PubMed, you get the title, abstract, PubMed ID number. As you scroll down the page, you get the mesh terms it's indexed with, corresponding to the terms in the tree I showed you on the previous slide, along with subheadings, if they're applicable. Subheadings tell you a little bit more about what the category it was indexed with is. And you scroll down further, some of these subheadings are chemicals that are also listed as substances. Arachidonic acids is an example, indoles. And then there are others too. These are the ones in lowercase, the one I've highlighted here, this compound, AM251, cannabinoid receptor antagonist. These compounds are yet another finer level of indexing. And these are called supplementary concept records in MESH. And these are where all the new terms that enter the vocabulary begin, where they're identified by indexers who index the articles. You search the supplementary concept record in MESH search function, you get a little more information about it. You have the name, a unique identifier number, and then the chemical names. You also have the MESH headings in the tree it's mapped to, the two groups here from the formula, pyridines and pyrazoles, the frequency or the number of times it's been used to index articles in the original source. So what is how are these new terms identified, and what determines whether or not these should be included in MESH? The process begins when indexers encounter chemical compounds, proteins, or microRNAs that are the main point of the article, but for which there's no term or supplementary concept record in MESH. The indexer then creates a flag for this term, which are reviewed by chemical specialists at NOM. They review these to make sure they meet certain criteria. Of course, if we already have it or have a certain group that would cover it, a broader group, and it doesn't yet warrant creation of a new record, then that won't be created as a new record for SCR. It may, however, be added as a synonym to an existing term if it's hadn't been encountered in literature before. For compounds, it's also the frequency has to be greater than 1. However, in order to comprehensively cover all gene products, even if it's the only publication of a gene product from certain model organisms or medically relevant organisms, even if there's only one publication, MESH will create a record, a CR for that. Some of the other criteria, general criteria for creating new terms for MESH, they have to be biologically relevant. This includes drugs, natural substances such as compounds extracted from plants, metabolites. They have to be, say, undergoing testing in organisms or cell lines in preclinical phase, or of course, in clinical testing or trial phase. Now we do make exceptions to the rule. If a compound is the only publication and it's in a clinical trial, MESH will create a record for that. Also, potential molecular probes or diagnostic aids, and they should have a descriptive chemical name or structure, which can be found in the article. For proteins and microRNAs, they must be from the 11 model organisms defined by MESH. These include humans, rats, mice, Drosophila, zebrafish, C. elegans, yeast, things like that, or clinically significant non-model organisms such as pathogens or microorganisms that may have a biological or say biotechnological application or for drug discovery. Say a good example of a non-model organism that's clinically significant, of course, would be SARS-CoV-2. And MESH has indeed created a lot of records in the past year for COVID-19, different drugs in the pipeline, as well as different gene products of SARS-CoV-2. So to show you in a little more detail what this looks like, here is what the report is showing the workflow when the new term entered the pipeline, when it was reviewed by the specialists. They had a comment to create a new SCR, that's the NCBI gene ID, for more information, and the new term that's recommended. Then the action of MESH for 3 of these new SCRs were created. As you can see what they map to. There's a mouse protein, a zebrafish protein, a plant compound, gibberellic acid, and then the one at the bottom, streptocyclinone A, was placed on hold. Now, why is MESH placing these terms on hold? Well, turns out for a long time, MESH was not placing compounds on hold or anything. And what happened was there were a lot of vague and ambiguous SCRs, which had very low frequency of use. Some of them may have been typos or were published in an obscure journal 30 years ago, and it got lost in translation. So we wanted to help declutter MESH by holding off on creating records for compounds with less than one publication, with only one publication, until there were subsequent publications of this. This helps improve the accuracy of indexing, as well as makes it more efficient by not having as many possibly ambiguous compounds for indexers to misidentify. But we don't want to discard these compounds because they could be very important awaiting clinical trials. Or this also gives us a tool to help track subsequent publications and citations in the literature for bibliometrics. And it could also be a synonym for a compound that's already in MESH, perhaps a different spelling that hasn't been mentioned in previous publications, but will turn up later. So this gives us a very broad coverage of the literature by placing these on hold, and allows us to hold on to potentially important terms. Here's an example of one such term, the MESH record for darcanelide. It was created in 1978 and hasn't appeared since then. So this was back 2 years ago, nothing was placed on hold. This still has not turned up since then. Will it? I don't know. You never can tell, but it's there. And by placing things on hold now, we don't clutter MESH in the near term, but we hold on to these things in the long term, for the long term, so that perhaps we can search for them later. So just to summarize the data from the past couple of years, there's been a similar number, about 3,500 of chemical flags reviewed. And about 90% of these are from single publications. You do get about 10% where you have more than one flag from a single publication. And about 60% of these are compounds, 30 to 40% are proteins, and about 2 to 5% are for RNAs. And now about half of these cases, new records or SCRs are created. 10% are adding new terms to existing ones or synonyms. And about 10% they're declined because they don't meet the guidelines, or they're not, we already have the term. Other actions include, it could be a request to change the heading map, the MESH term, to a more appropriate one. Now, you'll notice that a significant number, 25 to 30%, are placed on hold. And we've figured out a way to continue to search PubMed for these terms. In the left-hand column here, this was the original PubMed ID, and the term at the time when it was placed on hold. And researching, we find new publications of these that can then be created as new MESH records. Now, you'll note here, of these that I'm showing, 2 of them were not created as MESH records because we already had them. They were differences in spelling, perhaps, or we found that they were already in MESH. So not all of them that are placed on hold are created into new SCRs, but of the roughly 1,700 terms through August of last year, so far we found new publications that warranted creating a new record for 270 of these. And of the set, there's 1,359 remaining with only a single publication that we are continuing to search. Now, you'll notice these numbers don't add up here. And that's because some of the terms placed on hold, we reject and remove because they're very vague, say compound X or compound 1. You do see that a lot. If it doesn't have a description of its chemical action anywhere, we decide not to place it on hold if they didn't give us a handle to find any other publications of it. Now, the ultimate goal is we would like, of course, we're going to continue searching terms placed on hold with the set we have and new terms that are added. But what we'd really like to do is to develop an algorithm so that we can use machine learning to identify new compounds in the literature. And this will be very important as indexing moves towards automation. There won't be human indexers to identify the new compounds. So this is in development right now, and will be integrated with the automated indexing system in the future. So I'd like to thank my colleagues at NLM. Joseph helped develop the code for searching PubMed with the terms placed on hold, and everybody else on the chemical flags team, the chemical specialists, and also Justine Fitzgerald is a contractor with MESH who enters most of these terms in MESH's new SCR records. And I thank you very much. I will take your questions.
Thank you, Nicolas. So indeed, we have 2 questions from Charlie Hoyt that I will be reading to you now. So the first is, my experience with MESH has always been a bit tricky. It's ubiquitous even outside of the indexing usage you are describing. The issue I've always had is that it's difficult to figure out who is working on MESH and how to give feedback or discuss with them. Do you feel this is the case? How should people get in touch with MESH curators? Is there a list of people who are working on this and their contact information?
Oh, well, thanks for that question. I work on the indexing section, but know the MESH team quite well. And if you go to the MESH browser site, I can post a link for that. There should be an email for customer feedback, and also for submitting MESH requests for either adding terms or modifying existing terms. I will post that in chat. See the second question. Why does
yes, the second question is, why doesn't MESH map to other related vocabularies like chemical entities or biological interest from EBI?
That's a good question. MESH is part of UMLS, the Unified Medical Language System. That includes things like SNOMED, where some of the terms map to that. I'm not as familiar with its mapping with or interactions with CHEBI or other ones at this time, but that might be an area where they're moving to.
Okay, so thank you, Nicolas, for your talk. We are now going to move to the second speaker of today, who is Paula Duek, and she will have a talk on neXtProt function prediction community pages. So Nicolas, I kindly ask you to stop sharing so Paula can share a screen. Thank you. Thank you, Paula. Yes.
Yes. Thank you, everybody, and thanks to the organizers to allow me to present the function prediction pages in neXtProt. neXtProt is a database developed by the Califo Group that has affiliations to the University of Geneva and the Swiss Institute of Bioinformatics. So neXtProt is is a knowledge base for human proteins that contains data integrated from different resources, and it has also its own annotations. All the data in neXtProt is qualified as gold or silver, and gold is considered data of high quality data, and silver is data for which we have less confidence, but that it might be interesting to explore. Whenever possible, this data qualification is done in collaboration with the data providers. So neXtProt content is organized in different views, describing the different aspects of the protein. For example, here I show the function view, where we display summaries from SwissProt, Go molecular function and biological process terms from different resources, and pathways from KEGG and Reactome. In addition, there is a community space where there are tools developed by the community, and we have also additional tools developed by our group, such as the Sparkle Advanced Query. So for many years, we have been interested in proteins that do not have functional annotations, and in how we can help in their characterization. So the list of proteins without functional annotations can be retrieved by a Sparkle query that will give a list of entries that do not have SwissProt annotations or pathway annotations from KEGG or Reactome, and that do not have neither Go terms, biological process or molecular function Go terms, except those that are very, very general terms that are not very informative, just as protein binding or metal ion binding. This search also excludes entries with dubious existence they are not included. So the result of this query gives a list of 1,669 entries that represent 8.5% of the human proteome. 76% of these proteins have been detected at protein level. But although these entries do not have functional associated data, they do have other type of data, such as 3D structure, diseases, protein-protein interaction or expression. And for many years, we have been trying to combine this data to try to formulate functional hypotheses. So some of these functional predictions have been experimentally validated and published. Other functional predictions have been published and are in the process of being validated by collaborators in South Korea using worm as a model organism. Other predictions have been published and they are not validated yet. And some of others have not been published, and we would like to make them available. Some of these functional predictions are the result of a teaching experience. So in 2020, we started to give a course at the University of Geneva named Functionathon. This course is for undergraduate students in third year in biomedical science. And the aim of this course is to teach students to interact with databases and to interpret results from bioinformatics tools. And to do this, we use as a context a real research project and the aim of this project is to formulate a functional hypothesis for uncharacterized human proteins. So we have more or less 20 undergraduate students that are divided in groups of 3 or 4. They are attributed one uncharacterized protein entry, and they are followed by one tutor. Before the course, the tutors, they do an intensive literature search to verify that these proteins have not been, in fact, characterized but not annotated yet in databases. And if this is the case, we feed back databases. Then in the course, during 6 sessions, the students extract information from the literature, additional information, information from databases, and they run bioinformatics tools to try to collect as much as possible data. So to guess the function of the protein, they explore different aspects of the protein, such as they analyze if the protein, where the protein is conserved, in which organisms. They also explore if there are conserved domains, sites, or PTMs, if the 3D structure has been identified, or if it can be modelized. Also, they look for information on subcellular location, protein-protein interaction, expression, co-expression, phenotypes in model organisms, or variants, human variants with phenotypic description. With all of this data, they build what we call an identity card of the protein that has all the consolidated information. And we discuss with them the importance of keep high confidence data and also to keep the evidence for each of the observations that they are reporting. Then during 2 sessions, students work on the hypothesis formulation, and we discuss with them which type of that data can be used to directly predict the function, what could be used to test the compatibility of the different data. And we help them to formulate the hypothesis, analyzing that that they are in accordance with all the observations that they gathered. And we discussed that protein can have several functions, so more than one hypothesis is possible. Then in the last session, the students present the hypothesis with the evidences during a 10 minutes presentation, and we have a general discussion to see how this hypothesis can be experimentally tested in the lab. And then after the course, what we do is a standardization of those predictions and evidences using Go and Ecoterms. But this is done not by the students, but by the tutors after the course. This workflow and the results of the data mining of the first course has been recently published in a database journal. So in order to make available all these predictions from the teaching course and previous predictions that we have done, and also for all the community, we decided to do function prediction pages in neXtProt. So this is what, for example, one of them shows the predictions we've done for RSRP1. This protein has been data mined in the first course. This protein was shown to be conserved. We saw that it was conserved in vertebrates and that is broadly expressed, that the human and orthologs are predicted to localize in the nucleus, that is phosphorylated, ubiquitinated, and methylated in conserved residues across species, and that interact and co-expresses with CCNL1, CLK2, and 3 that are proteins involved in splicing and transcriptional regulation. And in general, this protein, RSRP1, it co-expresses with the nuclear functions. So here we translated the predictions using Go terms, and the type of evidence also are described here. And the underlying the details underlying those predictions are not displayed, but if there is a publication, we show it. This is the case here. And as we also provide the ORCIDs, this can be used to access for the detailed information. The ORCIDs could be the link between submitters and experimentalists that would like to test this hypothesis. So who is invited to send us functional predictions? All the scientific community is invited. And we have a special thought on biocurators because, in fact, much of this work is what biocurators do in their daily work. So we will be very happy to receive functional predictions from the biocuration community. Also, lab researchers are welcome. And we don't have a submission form for the moment, but you can use the contact option in neXtProt to send us the protein accession number for the protein that you are predicting. We encourage to send predictions for uncharacterized proteins that are the result of the Sparkle query. Send us also the Go terms, describe the molecular functions and biological processes, and submit as much as possible precise Go terms, not very, very general terms, but quite precise. Also send us the eco terms that describe the type of supporting evidence and the associated PubMed ID, if they are available, your ORC ID, and if you have published your functional predictions, also the PubMed ID. So here we have a page with that documents our project, and that also displays the list of entries for which we have functional predictions for the moment. We are working now on improving the functional prediction pages and in the submission form. So we know that for people not doing biocuration, converting free text into Go terms requires a specific training. So we are thinking that the submission form could include a free text for function prediction and for experimental evidence that could help us to check if the proposed Go and eco terms reflect really the functional hypotheses and evidences. We are also analyzing or thinking about how we could display the real evidence, the real data that is behind those functional predictions. So one option is to make available on our FTP site the free text for this evidence. But we are also thinking if we could use annotation extensions that combine cellular component Go terms with anatomy ontology terms and gene names, for example, for interaction, as I'm showing here. So with this, I finish, and I would like to thank all the neXtProt team for the hard work in all our projects. Feedback and suggestions for these pages are very welcome. So here you have our contact. And I also would like to thank all the databases and bioinformatics tools that allow those predictions to make possible, and the students from the Functionathon 2020 and 2021, and all of you for your attention. Thank you very much.
Thank you so much, Paula, for your very interesting talk. We just have time probably for a very short question, and I have a curiosity for you. So I'm very fond of training in biocuration, and we do it a lot also in our database, the neXtProt. Which was the approach and the reaction the undergraduate student had to such an intensive and very well organized course? Did they develop a further interest in going on with biocuration, having further training, maybe in learning how to convert the free text into geo or eco terms?
Some of them were interested, and they are trying to do these conversions. And then that's where we really realized that it's not evident. Others were interested, and they asked to repeat the exercise with new proteins. So but the scholar year is finished, so we did not do, but they were really motivated. Well, so we have everything.
I see. Thank you so much. So now we have the first speaker of today, who is Yasmin Young, and she's going to talk about PDB biocuration on the front line of structural biology. So thank you, Yasmin.
Thank you. First, I would like to thank organizers for giving me this opportunity to introduce biocuration at the Worldwide Protein Data Bank. PDB is the first open digital data resource in all of biology. It was founded in 1971. PDB is a single global archive for 3D macromolecular structure data. It adheres to FAIR principles. Worldwide PDB is an organization that manages PDB, EMDB, and the BMRB 3 core archives. The members of the Worldwide PDB are RCSB PDB at the Rutgers in US, PDBJ at the Osaka University in Japan, PDBE at the EBI in UK, BMRB at the University of Wisconsin in US, and the recently newly joined EMDB at the EBI in UK. RCSB PDB is the manager for the core archive, PDB core archive. We collaborate on deposition, biocuration, and validation. Each partner provides complementary website to serve PDB data with different views. The key to long-term success and the sustainability is forming the Worldwide PDB partnership. Currently, we have 15 biocurators worldwide working at the RCSB PDB, PDBE, and the PDBJ. We maintain a single global archive, no matter which PDB site users download data files from. We ensure identical data files are provided. Worldwide PDB biocurators use a unified one-step system to curate data. These systems provide geographical workload distribution, as you can see on this map here, that RCSB PDB share 47% of the workload, while PDBE shares 31% of the workload from Europe, and the PDBJ share 22% from Asia. Biocurators set up standard procedures and published these procedures, follow these practices to ensure data standardization among PDB partners. We also review PDB archive and bring the data up to the modern standards by conducting remediation effort. Why is biocuration important at the Worldwide PDB? We manage data deposition, quality, and integrity, and provide integral support to the research community worldwide. To ensure the data are fair, we focus on data standardization and quality control. For data standardization, each PDB entry is assigned a unique and persistent identifier. Data files are brought to semantic integrity and a common format that permits large scale data analysis and distribution. For data quality control, we cross-check with external database references, such as UniProt, NCBI, or PubMed. Whatever possible metadata are conformed to control vocabularies, and we set range limits to catch data outliers. What do we biocurate? How do we biocurate at the PDB? At the deposition time, biocuration is provided by data depositors on the sequence and the taxonomy for polymers, and the chemical information on ligands if authors' ligands do not match to the PDB chemical component dictionary. After data submission, Worldwide PDB biocurators cross-check authors' sequence and taxonomy with external references. We assign existing ligand or add new ligand definitions based on author-provided chemical information. We also provide secondary and quaternary structure as value-added annotation. More importantly, we validate structures against experimental data adopting community recommended validation software. And we provide depositors an official validation report for their manuscript submission, which is now required by many journals, such as Nature, Cell, Science, Structural, JBC, etc. You can find more information about our biocuration at the paper that is published at the database journal. Throughout the COVID-19 pandemic, worldwide PDB biocuration staff have continued to support research, training, and education worldwide. All biocurators have been working remotely since last March. The use of unified one-step system has made this possible with very easy transition. We have record high number of depositions with SARS-CoV-2 structures accounted for 7% of the depositions last year. For the benefit of global health, as the structural biology community step up to provide much needed insights into SARS-CoV-2, we have prioritized the SARS-CoV-2 structures and working very closely with depositors to release their structures immediately. As of August 1st, 2021, more than 1,400 SARS-CoV-2 structures are now freely available from the PDB, reflecting enormous efforts made by the structural biology community in fighting the pandemic. You can find out more COVID-19 resource at the rcsb.org/COVID-19 for easy access of those structures and educational materials. We have received more than 1,800 SARS-CoV-2 structures as of August 1st, 2021. Among these, more than 400 batch depositions are coming from fragment screening study. We are thrilled that our efforts have contributed to general awareness of the importance of scientific advances. These structures are contributed by 234 principal investigators from 20 countries. The median time to complete biocuration and release this data is about 36 days. It was 14 days at the beginning of pandemic when there is an urgency to make the data available for the benefit of global health. You can find the rest of the information at our website, rcsb.org/COVID-19 for those resources. The very first structures on the SARS-CoV-2 with PDB ID 6LU7 was deposited on January 26 last year. It was quickly released in working with depositors in about 1 week. PDB has recently provided coordinate versioning to allow depositors to update their released structures without change of PDB ID. Occasionally, rapid PDB deposition and the publication of SARS-CoV-2 structure studies driven by an understandable sense of urgency has resulted in public release of PDB structures containing minor errors. The worldwide PDB coordinate versioning feature has enabled rapid correction of SARS-CoV-2 related structures archived in the PDB. To make the data findable, SARS-CoV-2 structures were updated with standard molecular name, taxonomy, and EC number when UniProt references became available. we also provide COVID-19 outreach. We have many educational materials, including posters, paper forwarding, and the videos. Those are available at the rcsb.org/COVID-19 that you can explore. I also would like to let you know that this year we are celebrating PDB 50th anniversary with many events and outreach materials. You can visit rcsb.org/PDB50 for more information. We have PDB 50 games that you can explore the process of structured discovery, and also playing cards that with many different structures on the cards. And I would like to thank you, and I will be happy to take any questions you may have.
So thank you so much, Yasmin, for a great talk. I have a couple of curiosities for you. So first of all, it has been a great initiative to prioritize SARS-CoV-2 structured annotation in the database, since due to the global pandemic, that was the most relevant aspect to Biocurator. So, of course, this called for a much greater effort in curation. And you said that all the curators have been working remotely. Have you also been introducing new curators to the database who got interested in during the last year, during the pandemic?
Yes, we recruit 2 new biocurators during pandemic. It was quite a challenging, especially one came from Poland. And so what we have done was we immediately discussed with all existing worldwide PDB biocurator, and we revised the training procedure to allow remote training. And we use Zoom for the training, and we provide a training for 2 months until we feel safe to bring this new biocurator to US. And that's what we have done. And then, of course, we have been remote working since last March, and we are still remote working currently. And we continue remote training this new biocurator in United States, even though he has moved to United States, still continue working remotely. And it has been working quite well. I have to say the most challenge that I gather from these 2 new biocurators is the anxiety they have without being able to be part of the team and have a face-to-face communication. So based on that feedback, we then provide social hours. We have weekly social hour to bring these new biocurators, make them feel to be part of the family. So that's what we have done. Yeah.
Thank you so much. It's really impressive, and it's difficult and a challenge to deal with training new curators during the pandemic, but it's great to do it also on remote. And now, thank you, Yasmin, for your talk. And we are going to move to the last speaker of today, who is Anna Taska, she's going to talk about the DrugMecDB, a database of drug mechanism. So thank you, Anna.
Okay, thank you. Let me share. Okay, can you see the first slide?
Sure. Yes.
Oh, great. So hello, everyone. Thank you to the organizers for inviting us to present this work. I'm very pleased to present today a DrugMecDB, a manually created database of drug mechanisms. So in the field of drug curation, we can definitely distinguish databases of drug targets. And here are some examples like Drug Bank, Kembo, or Inside Drugs. So from this example of imatinib drug, we can quickly pick up that one of the targets of this drug is BCR-ABL fusion protein. Some of these databases have also section on drug categories, like central nervous system depressants, which gives us some hint about drug mechanism of action. However, there's so much more information in the free text summary. This is very high quality data, regularly updated, but the main pitfall of that free text is that this information is not computable. So to address this issue, we decided to extract drugs mechanism of action information and present it in a knowledge graph form. And here I'm going to show you how we created graphs from this free text mechanism of action description. So this is mechanism of action for drug fomepizole that treats methanol poisoning. So we extracted key words such as drug name. We know that it inhibits alcohol dehydrogenase, which catalyzes methanol to toxic metabolites. So from this information, we could draw a knowledge graph that consists of the concepts like drug or protein or biological process, and it consists of relationships that connects those concepts. So for those who are not familiar with graph theory, I should mention here that what is represented here by a circle in this graph, it is called a node, and that what is represented here by an arrow, it is called an edge. And this will help you to understand the format that was used for the graph generation. So in our graphs for nodes, we use concept types such as proteins taken from UniProt database, or biological process taken from gene ontology, and chemical substance, disease, and drug were taken from MESH database. But we use so many more ontology sources for our concept types than the one I just presented in the previous graph. And for chemicals, we use KB. For gene families, we use InterPro and PFAM. For anatomy, we use Uberon. Cell types were taken from cell ontology. Phenotypes were taken from human phenotype ontology. Pathways from Reactome, and taxonomy was taken from taxonomy browser and CPI. So to describe relationships between nodes, we use BioLink model, which is a high-level data model of biological entities and their associations. So in more details, we use BioLink model for the BioLink classes such as protein or a drug, but as we also use BioLink model for the predicates. So predicates helped us to describe the relationship between nodes in these graphs. So to represent graph information, we use YAML format. And this is because this format can be easily consumed by programming languages like Python. And each YAML record consisted of several components. And first was the component that provided us an information about the graph, like unique graph ID, disease name, disease identifier, and the drug name and drug identifier. So nodes contain information on each of the concepts in the graph. So each node contains the field ID corresponding to the external identifier, name of the concept, and the label representing a concept type. Links represent the relationship in the graph. So each link contains the identifiers for the source and for the target concepts, and the key field is here for the relationship type information. So links were ordered the way that the first source is a drug identifier and the last source is a disease identifier. We also included reference section at the bottom of the YAML file. So this is an output of the described earlier YAM file, and this is a simple path with detailed information on how this drug, which is an antihistamine drug, treats vasomotor rhinitis. And some drugs produce their treatment effects through multiple interactions occurring at the same time. And this may include the inhibition of multiple targets or acting on multiple unrelated pathways. So these processes can be represented through a branching path, like the one presented here on this slide. So this is a graph that shows how metformin treats diabetes type 2. I just added this picture of this graph to show you the range of complexity of the graphs in our database, but I need to add that these complex graphs are relatively rare. So this is still work in progress. So far we curated over 1,300 drugs, over 600 diseases, and almost 3,000 unique drug disease combinations. And based on our estimate, we are about to, we can about curate 4,200 drug disease combinations. So here are some overall trends in our database, and the most common node appear to be biological process, followed by protein, drug, and the disease. And the most important edge in drug MACDB is drug decreases activity of a protein. So by looking at these statistics, we could perhaps say that these are the types of edges that we should be spending more time, more curation efforts on. So this is our website. This is the address. You can access drug MACDB. It's a free access website. You can search by drug or by disease. There are tables with concepts and relationship on this website, and you can also access a GitHub repo for our project. So if you'd like to contribute, drug MACDB contains detailed curation guide information. So just click overview, curation guide, and you'll find all necessary information on how to perform this curation. So at the end of this talk, I would like to cover briefly why we think drug MACDB is important. So our team is planning to use drug MACDB in some of the computational drug repurposing work. And drug repurposing is the use of existing drugs or drug candidates for new disease indications. And potential benefits are reduced time and reduced costs in drug development and less uncertainty related to safety and pharmacokinetics profile. So I have to say that there are many strategies to identify drug repurposing candidates, but we are mostly interested in knowledge graph reasoning approaches that utilize large biomedical knowledge graphs. And this is because these approaches are most similar to what human scientists do in creating a hypothesis. So as one example, let's look at imatinib, which has long been known to treat CML, chronic myelogenous leukemia, and it treats CML because it inhibits that BCR-ABL fusion protein, which is part of CML pathway, which causes CML. And also based on literature, we know that imatinib inhibits c-KIT, which is important for the mass degranulation, which causes asthma. So you could make this hypothesis that imatinib actually treats asthma. So this was actually a hypothesis that scientists came up with, and they actually tested it in a randomized clinical trial, and they found out that people treated with imatinib had less airway reactivity in imatinib-treated individuals. So the idea of combining drug repurposing with knowledge graphs is gaining popularity. Many more people are getting interested in this approach. So we think that drug MACDB can be very useful in this type of algorithms, either as a training set or evaluation set for knowledge graph reasoning algorithms. So here I would like to thank you to all curators involved in this project, as well as thank you to Andrew and his team for this amazing opportunity and for the support they provided. And thank you all for listening, and I'm happy to take your questions.
Thank you, Anna, for your great talk. I have some sort of curiosity about your
Yes.
database, as you do a great effort in covering several aspects for what concerns drugs, diseases, and the correlation between them. Which are the main challenges for you in biocuration of drugs, diseases, and the integration of all this information in DrugMacDB?
The most challenging information, it was, first of all, now all drugs have clear mechanisms of action. So describing which mechanism of action is a set mechanism of action that we can add to our database, and which mechanism of action is actually a secondary mechanism of action, which is not yet established and confirmed by clinical trials.
I see. Thank
you.
Thank you so much. So I think that we are now at the end of today's session, of this third session of the ISB 2021 virtual conference. So thank you all for attending, and we really, really appreciate it. And you know that we are going to have the next session, which is going to be in October, the 5th of October, and it's going to feature the annual meeting of the society, and also the a poster session. So please stay in touch. We are going to share more information with you soon, and you can also look at the ISB website for all the news about the upcoming session of October. You know that today's session has been recorded and is going to be available in YouTube in the next few days. And please keep on submitting abstract for October session, since we are really looking forward to receive your abstract for poster and talks. And with this said, I would really, really like to thank today's panelists, speakers, our chairs, and also all the people who have been volunteering in organizing the session and the conference. So thank you all. And we are going to see you again October 5. Thank you so much. Bye.